So you use plants and microbes to pull heavy metals out of soil and water. If a friend asked you what you actually do all day, how would you explain it?
Yeah, see, first of all, we need to know the heavy metal pollutions, especially throughout the world. It has become a threat that's not known to everyone. Because when we quantify, like we are quantifying soil, or we are quantifying water, we are quantifying drinking water, if we find the amount of the metals in it, it becomes important for us to deal with them. Because the physical options what we have available are cost benefits, and the health hazards associated with these metals is really big. They are very much detrimental as far as human health is considered. Keeping this in mind and having one of the important medicinal plant from Kashmir Valley, that is saffron, that's being transported, exported throughout the world, it was impacting this plant. And we were seeing that the DNA damage was incorporated in this medicinal plant. And it's also used as spice in Jammu and Kashmir and India as well as outside the country. The initial stage was when we confirmed the DNA damage that is causing to this saffron, its Crocus sativus. And the second was what is the possible regions. So we profiled the soil, we got to know that there are a number of heavy metals, especially if you talk of mercury, if you talk of lead, if you talk of cadmium, if you talk of nickel, they were in the good concentrations. But what we find is that the metal concentrations for the plant was above the normals. So we try to find out what can be the biological implications, how we can use microbes as well as plants to detoxify or to reduce the impact of metals, and especially mercury as well as cadmium, lead, and nickel. So we'll first screen out the metal tolerant bacteria because there are a good number of bacteria which are inhibited to soil. But every bacteria cannot be tolerant. We have to screen out the tolerant bacteria from the various environments. Since having a site which was already quantifying the amount of metals, so the chances of getting metal tolerant bacteria are also high because they live in that environment. But what the concern is that they are under stress. They are not able to perform. So we screened a good number of bacteria. I think in my thesis, we have total screened of 72 bacteria in total, different species of bacteria. Bacillus, Pseudomonas, Klebsiella pneumonia, other bacteria, which we identified. And after that, we give their in vitro lab test. We check their resistance for a wide range of the, we started with 10 ppm, 20, 50, 40, 100, according to the range. And what we find where we got a good number of bacteria which can withstand with a good number of the concentrations.
Ye-
So when we got the metal tolerant bacteria, then we what we did is we try to use this again these bacteria to see how much concentration they can stir in 48 hours. Because now we have a clear picture that these are the bacteria who are the metal tolerant bacteria. Because for the metal tolerance, there's a mechanism. What we call, we can see the shield mechanism. When the bacteria are put to the pressure stress, there are some hormones, there are some enzymes which are released by these bacteria in order to tolerate that stress. So they become tolerant bacteria. Now, from the tolerance range to the excretion range, we again did the experiments, in vitro experiments in the lab, to check that how much of quantity they will be able to sequester, they will be able to take. Because bacteria as well as plants, they have the quality that they can take the metal inside them. There are number of systems which are associated. Efflux system is one. If you talk of plants, they have the roof, the xylem phloem system. And on the behalf of the cation exchange capacity, there is also exchange of the metals. If it's a divalent and trivalent, based upon the cation exchange capacity and the ions, they exchange. And what we found that we got a very good number of bacteria which can really stir the metals, especially lead, mercury, cadmium, and nickel. This background, we choose this problem to find out the solution that is environmental friendly.
So your 2022 paper calls a plant called coontail an accretion biotool, which this plant is also pretty commonly known as a weed. And Kashmir has been pulling it out of lakes since the 1980s because it's a bit of a nuisance. So what made you look at it as something people pay to get rid of and study it instead?
Yeah, see, Kashmir, the Indian part of Jammu and Kashmir, what we see, we have got a very good number of water bodies, especially in terms of the lakes. If you talk of Asia's freshwater lake, it exists in J&K only. It's Waller Lake. And this plant with Ceratophyllum demersum you are talking about, we used it as a practical plant. Reason being, we checked every plant which was aquatic there. We checked their potential to take up the metals. So again, we did an in vitro testing. We did it in the lab. We got the plant. We nourished the plant. We gave it same environment, that kind of a lake environment what it was. And then we stressed it. Then we put the metal concentrations so that we can see what was the initial concentration in our laboratory and what when we put the plants after that. And continuously we were taking the samples after 6 hours, 12 hours, 48 hours. And then we see again, this plant has a very good capacity to stir the heavy metals. This was not a selective one. We took good number of plants. Like we have Salvinia, we have Tropa, we have Ceratophyllum demersum, we have Nilambu nucifera. We tried this experiment on the other plants also. But the successful trial was conducted on this plant, Ceratophyllum. So we try to explore the opportunities where we can use, because this plant is not confined to only Indian scenarios. It is also confined in some of the Western countries. Throughout the world, it's available. So if the problem exists throughout the world, then this plant has a solution, and that's an economical solution, eco-friendly solution we have.
Yeah. So I also interviewed a man named Bruce Rittman, who actually won the Stockholm Water Prize for engineering microbial communities to clean water, and he builds the systems from scratch. And you work with what's already growing in a lake. So does one of those ideas scale better in a place like India, or are they solutions for different problems?
We conducted an experimental lab. When we come to the natural conditions, we need to integrate several technologies. The conditions what I am giving to a bacteria in the lab are different and what can be in the natural environment. Not only one bacteria can do it, consortium of bacteria can do it. So we need to integrate science as well as technology, which can be used, and we can flourish this concept. Again, in India as well as outside the India, if it flourishes, if we are able to make a product based upon the consortium, then it will be a product, it has a marketable value there. We should come on. See, nowadays we are with the concept of biofertilizers. Again, we are using the bacteria, consortium bacteria, having some certain of ability. If we are able to transform this ability, the sequestration capacity of the bacterium consortium, and if we come up with some machine like a composter or something like that, it will be a very great invention, or it will be a very great helpful for the community, especially who are looking for the address the environmental issues.
Yeah, so can you give me one concrete number from your results, which can be a concentration, a removal rate, something that you think is really easy to picture?
I will share you one of something with you. If I give you the summary of the work which we conducted is, it's all about molecular levels. I will tell you the numbers which was more evident: inhibitory concentrations and sequestrations. This is actually the number which I'm trying to recall. If I talk of this number, Pseudomonas putida, and it's already submitted in NCBI, and you can check it on NCBI website. It's with a molecular code. It has a highest potential of 73% of mercury after 48 hours only. So this Pseudomonas putida, which was we isolated from metal contaminated sites, and it proved to be a beautiful species in terms of expression. 73 cadmium, if you are going to sequester from that, it's a big number. I think the number of 48 hours, if you have a cadmium concentration, that's in vitro testing, and 72% of its biospecies, it's itself a success. Same
Yeah.
if you talk of other species, that is Bacillus thuringiensis, it again shows a good result. See, removal efficiency of this, it is reported several species have been reported. It's not only first stages we are reported, but for lead, I will tell you, there was a species. It is published in one of the scientific papers. You can access them from the Google Scholar. This was the first report throughout the world of the lead regarding the sequestration potential. And that's why we not only confined it with molecular identification. If you will see the papers, we have done the identification not only based on the molecular, we have done the MALDI-TOF analysis, proteomic analysis of that in order to confirm the species at that time.
That's a very, very interesting fact, that percentage of those heavy metals. So right now there's a lot of enthusiasm for phytoremediation as a green cleanup answer. So what can plants realistically do there, and where is there a little bit too much hype?
We need to first identify them. I will tell you a plant which consists a good number of heavy metals, Brassica rapa, mustard. If you have a site which is totally destructed, it is impacted with the heavy metals. Do one cultivation of Brassica there. It will give you clean because Brassica has that capacity that it can stir everything what is in the soil. But the second factor regarding this plant is that it does not only filter heavy metals, it can take everything from the soil, basic nutrients, micronutrients, macronutrients. So what actually we need to see is that first is if we tell on the lab levels, we have to check their plant's efficiency, and what impacts it is going to have if we use a particular plant. Definitely phytoremediation, bioremediation, or microbial remediation, they are going to help us in a greater way, in a broader concept. But the thing is that we need to first select the plant, check the efficiencies, then to come on the practical aspects.
So you went from Kashmir to Sonepot in Delhi NCR. So those are two with completely different environmental problems. Did that move really change how urgent you think the situation is in those places?
See, why I shifted from Kashmir to Sonipat is because of academic shifts. Working as an head of the department in SRM University, but problems still remain the same. If I talk about the heavy metals, a good number of heavy metals are again in the soils and water bodies of this place also. Why? The reason is that if we talk about the agricultural practices, especially throughout the world, nowadays we are more dependent on the pesticides, insecticides, and other things. And these pesticides, these insecticides have a good amount of the heavy metals which they consist of. So we are not getting out of the problem. The population is growing up. We need to feed up the populations. We want to grow more. We want to produce more. So in this race of production, we are degrading our soils. Our soil health is going down because of the intensive use of the pesticides. And in the pesticides, a good ingredients is the heavy metals. Here we have also conducted that it is in the initial stage, the screening of the soils as well as water. And what we find is that, yeah, this place is not also the exceptional. A good number of heavy metals still exists in the waters, in the soils, in the groundwaters of this place.
So on a scale from 1 to 5, 5 being the most urgent, how serious is heavy metal contamination in India's soil and water right now, and why that number?
I will try to pick it on power four, scale of four. You see, the health impacts of this are totally disastrous. It's a silent killer. Knowing that I'm taking a water, it has a low concentration of XYZ matter, but it's bioaccumulating. A time will come, my body will not allow me to digest anything. We are taking a slow poisons. We are giving our generations a slow poison. I'm giving the cereals, the crops we are producing from India or throughout the world. We don't know the concentration of metals. That means we are putting our lives in great danger. So I will put this problem at 4 out of the 5.
So who are a few scientists who you think I should interview after you? They can be Indian or any from anywhere.
Some of the scientists are from MIT, Massachusetts, they have done a very great work on that. Besides, if you go for India, one of the scientists is from the University of Kashmir, Professor B.A. Ganai. If you want his address, I can mail you that address. He's a really a great scientist, not only science, he's actually an environmental biochemist. So he has worked on this issue also.
I'll send you an email afterwards with all the information. I have one last question, and I ask every scientist this question. It's if every teenager on Earth had to hear one final sentence from you, what would it be?
Stay friendly, especially use environmental friendly products. Don't rely on the chemical fertilizers at straws. Be natural, try to be organic, use organic foods. If you save lives, that's what is important. Money does not matter. What matters is a good environment. What matters is the life. If you have life, we will come across to new inventions. we'll get everything what we want. But once if health is lost, we will not be able to do something better for the society, I think.